Warm welcome to all for coming today. First of all, I'd like to tell you who we are, who are the participants in today's session. It's about cell and gene therapy, what we do at EIC. I'd like first to thank our three guests, our three keynote speakers who are with us today. Professor Hans-Peter Kim, Professor Seppo Hertoala and Dr. Michael Winger. I'll say one more word about their vast experience that is very useful to us as we're moving along. We have with us today 9 Pathfinder projects that were selected and are now they're working together. And I'd like to thank all the representatives of those projects that are here with us. But this event goes beyond the 9 Pathfinder portfolio projects. It goes and covers, we call it the family of EIC, the family of cell and gene therapy, meaning projects from our other 2 financial instruments, our 2 stages of development, I should put it even better, transition and accelerator. So we have more than 20 representatives here from over 20 projects. Almost the entire family of cell and gene therapy from EIC is here. And of course, we have with us representatives from EIC. Stefano Aki is with us, who is the head of big department, and other heads of units and colleagues here. And Keith Sequeira from DGRNI is with us, and we'll give his welcoming remarks in a second. Having said all that, I'd like to now share with you why we organize this and what this session is about. But first of all, a big thanks to these 6 people. I hope I learned something from my colleague Cornelius, who always starts by saying thanking the people who contributed. So thanks Yanis, Penelope, Juliana, Gonzalo, Vilma, and Robert for the work to make this happen, because it might be an hour and a half, but it's not easy. Take my word for that, to really put these things, and they work hard for that. So. What are the objectives? What is the purpose? Why we're doing that here? We're doing that in this conference because we want to inform specialists of what we do at EIC in cell and gene therapy. We also want to inform public. This is also important, and the slide comes very soon to give you some more data about that. But very important for us is to demonstrate why we think that there is power in this portfolio, in the cell and gene therapy portfolio, from the way it was conceived, created, developed, and now it's running, and we're managing it. And finally, that's why we have the reception after the symposium. So we have ample time for networking and discussions between you and our advisors and the people we have invited to make it really interactive. So unfortunately, there isn't going to be questions here. Perhaps in the end, we may have some questions, and John Hodgson will take care of that, who is will be moderating the panel. But we have a lot of time later on the reception for discussions and networking. So what is the EIC Pathfinder? You will have a brochure that has been distributed, so I'll move to the next point because it's all there. You can see the 9 projects that we have put in this paper. It's for you to know what this portfolio, Pathfinder portfolio, comprised of. 9 projects that have been divided for scientific reasons, for reasons that can work together, to 2 sub-portfolios. Now, those are the names, but I will skip the slide because, as I said, you have it with you. The next point is why we created this portfolio. We created this portfolio on the basis of challenges that exist in the cell and gene therapy area. That was the beginning, that was the original conception, which are in the safety, in vivo efficacy line. Our first keynote speaker will address, of course, the issues at the clinical level. So we focused on the third point, which is manufacturing scaling up, which is at the preclinical level, given that this portfolio is at the Pathfinder level. So we focused very much on scaling up and manufacturing. But the reason that we started working is, of course, the multiple and complex challenges that are in this field, which has been going on for 30 years, but some revolutionary things are happening in the last 3 years. Some of these reasons are described in this slide and listed here. The lack of maturity in manufacturing techniques. I don't want to get into details. process standardization, scaling up, cost effects, and the last point that you see, which I have highlighted as green, the lack of innovation in these systems. And that was for us very important. And that was back in 2020, we started thinking of building such a challenge, EIC challenge, the outcome of which is the portfolio. But how we did that, we didn't know at that time. We had to really invent a model. We had to really invent reasons how to develop and how to work and how to select our projects. And Professor Hirtuala, he was a member of the committee, and he will share his views about that later on. And what we did is that we thought that in order for the projects to want to work together, they have to share research interest and research development interest. I don't want to call it business, but research development interest in their field. And that is how we selected the projects. And in the end, we realized it would be more efficient and effective to have 2 sub-portfolios within the 9 projects, 6 and 3, one focusing on the cell therapy manufacturing and the other one focusing on gene delivery systems. Again, the projects were selected. First, they were evaluated, and the top-ranked projects were selected based on the laws maximizing the common element between them, which would assure that they really want to work together. And this is now being proven, as you will hear later on the discussion, the panel discussion between projects moderated by John Hodgson, who is a former head of Nature Biotechnology and currently advisor of Nature Biotechnology. What are the 3 targeted areas of action at portfolio level? Is what you see here: technology, regulatory, and exploitation. And working at these 3 different areas, the targeted areas, of course you tackle common challenges, but you also leverage opportunities for working together. And in the end, you make the distinction between individual project work and portfolio work, and you expect the aspiration is that the work at the portfolio level will be more than the sum of the individual projects. And it is like that. How do we work at portfolio level? And again, you will see it live in a discussion moderated later on with 5 representatives, from 5 different projects at portfolio. This is just a schematic representation of the interaction between the projects at portfolio level from the first sub-portfolio. There's no time to get to details, but you see that Project A interacts with Project B on a specific objective, and then activities are organized in order to meet this objective. This is the logic behind that. And because these projects, they share research interest, that makes it a workable model. And this is the second sub-portfolio. You see 4 actions here, 4 objectives, and activities have been designed and are executed, starting executed right now. We are describing all that in a strategic document, strategic and roadmap document of which includes the strategic plan, the work prior to that, and the implementation of all the plan that will be published by EIC in the coming weeks. We didn't stay at the level of the Pathfinder portfolio, and along the way we realized the importance of gathering all the projects and companies in the cell and gene therapy funded by EIC, and that's what we did. And we did that, I believe, for the first time. And these are projects that focus in the same niche area within the cell and gene therapy, and they're all invited for this event for obvious reasons, networking reasons, to listen to our advisors, and to create, I don't particularly like the word community, I prefer the word family, but to be all together, that we're in the same field funded by EIC. And a reason that we invited also projects from the accelerator is here. We have a very successful accelerator company, Sparing Vision, that was funded by us, of course. Then they raised funds from private VCs. They are extremely successful, and out of the latest successes of this company, again, is an EIC fund accelerator company. They struck a deal with Intellia Therapeutics, which is one of the most successful companies in the US right now, running clinical trials. And this is, we think of that as quite a success. So there is a very good reason to have all the projects together to transfer this interaction. It's a very targeted interaction and a very important one. Finally, you can see here major investments, over 100 million in the exact areas that the EIC cell and gene therapy portfolios fall subject-wise. You have the RNA therapies portfolio transition level, and you have the Pathfinder level. Those are, what you see here is from January 1st to May 1st, 2023. And we conceived and made this challenge and this call 2 years earlier. So it's booming. It's I'm going to skip this slide, but Professor Benning, this is to see a little bit the other side of the side that you have to communicate the society, and this event plays that role as well. Professor Benning, I think she's one in our consortia. Ioannis, if you could remind me, I think she's a member of a consortia of the Pathfinder portfolio, and you see what these people say, and I believe what we do today, it's in line with the need to communicate, to have the stakeholders together to inform. Final comment is about why we think that with this portfolio we are positioning EIC quite well strategically, and we believe that EIC is at the right moment and gaining a really competitive advantage. That is my last slide. All these lines of evidence here. All these things happened in the last year that you can read. The biomanufacturing initiative from the US government was launched, I believe, December, 8 months ago. We're going to have a lot of activity in the biomanufacturing field from the Commission next year. Bayern just bought some facilities in California, and they are building $250 million plan in California to produce cell therapies, so focus on cell therapy manufacturing, but that is exactly what is our first sub-portfolio. Novo Nordisk just announced this plant was going to be operational in 2025, major investment in cell therapy manufacturing area again. You see this, this came in the news the other day, Harrington Discovery Institution, investment of 200 million in projects at the in Oxford University, and they expect to develop therapies for rare diseases in the years to come. So all this development has happened in the last months, focus in the same area that we have our portfolio, and because we were there 3 years earlier, we believe this is important to predict, and it does really give to the EIC competitive advantage. I'll stop here, and I'd like to invite Keith for welcoming remarks, please. Keith Sequeira is the head of unit. Unit's called EIC at DGRNI.
Thank you, Danos. Great pleasure to be here. I've been given 2 minutes by Danos, so what I'll try and do is to set a little bit of the context about what the European Innovation Council is, that this is embedded in. Some of you may know it, but I'm guessing a lot of people here are not so familiar with the European Innovation Council. It was launched by the Commission just over 2 years ago. We think it's quite unique. It's unique for Europe. It's very different from what we've done in previous framework programs, the Horizon 2020 program, for those of you who are familiar with it. And we also think it's quite unique in the world. So what we're able to do with the European Innovation Council, which is super relevant and is making possible what Danos described, is that we are able to support projects from very early stage research, breakthrough technologies. We are able to fund translational research. This is our transition funding. We are able to support spin-out companies, startups, and we are able to put significant, not only grant funding, but investments of up to 15 million euros in these startup companies. We're focusing on deep tech because that's where we see we need to accelerate this path from breakthrough science to application. And that's also why the areas that we see it's very hard for investors to come in at the early stage with the kind of risks that are involved. So I think for cell and gene therapy, this is one of the areas that we're focusing on. And the second thing I'd like to highlight is what Danos was talking about with the portfolio approach and the active management of this portfolio, which is very new for us in Europe and the European Commission. What we've been doing, we've been hiring program managers. Iordanis was the first program manager that we hired in the European Innovation Council. And their job was very different. Their job was not to check cost statements, to run administrative processes. Their job was to focus on strategy, identify the areas that are important where science is on the cusp of bringing innovations, applications, and then give them tools to actually develop this area proactively. So what Danos has described to you is the first of our, I think it's one of our first portfolios, cell and gene therapy, where we called for projects, but we selected the projects based on a portfolio. So it was not the standard peer review, and then we fund projects in isolation, which is what we've tended to do in the past, but deliberately to have a cohort, a portfolio of projects, and to identify where are the points where these projects can interact, collaborate, compete. As Danos was saying, I think in this area, one of those areas which is super important is manufacturing. But also, we can bring in here the ability to bring together the kind of research phase with the translational phase with the commercialization phase. So we have a growing portfolio of startups that we're funding across a number of areas, but already some of the ones. And what we think in many of these areas, but particularly cell and gene therapy, is that the pathway from the science to the market is incredibly long. There's lots of things that can go wrong in the process. And as public funders, we've tended to be too segmented. So we have one program funding the kind of research, and then we have another program funding the translation, and then we have another program funding startups or SMEs. And actually, this adds a lot of delay to the process. You have to then spend several years looking at your next stage of where you take it next. So what we're really trying to do as well is to reduce this and to make it much smoother to go from the research phase, translation, and commercialization. So maybe that's enough. I hope it's useful just to sort of set this in the context of what the European Innovation Council is trying to do generally, and also why we think this is super important for the cell and gene therapy area. It's important for Europe as well. We're really trying to focus more on the strategic technologies of the future. The European Innovation Council is very much at the forefront of this effort, and we've already identified some of the key technologies that we think Europe really needs to invest in, and biotech, and within that cell and gene therapy is certainly one of those areas. So this is the first time we've had this kind of meeting, bringing together a portfolio, the first of our portfolios, super important area. We're also here to learn how well this is working. We've seen that it's really, I think already it's been impressive what's been achieved, but looking forward very much to discussions and to see how this approach works and how we can make it better. So thanks very much.
Thank you, Keith. I'd like to invite Stefano Eki, who is the head of a very big department at the EIC, to give his welcoming introductory remarks.
Yes, thank you, Danos. So I'm the one who actually is trying to bring together this different funding stream that Keith just introduced for you at the level of the European Innovation Council. So we are trying to bring, early stage research in disruptive technologies on the Pathfinder, an instrument that we have designed to bring those technologies that are emerging from the Pathfinder to the moment that they can start their journey towards commercialization. And then finally, with the EIC Accelerator, we have an instrument that brings together grant support for the remaining R&D costs that we are facing for these projects, and also help startups to get, the necessary equity in order to start their journey on the market when they are trying to get funding from private investors. Here what you had a very good example that was exemplified by Danos is our ability to actually concentrate throughout the chain of funding on those, promising technologies, and being able already at the level of the accelerator, since we have many projects already in the area of cell and gene therapy at the level of the accelerator to support companies that are already at the stage of commercialization, some of them already raising funds with our support in order to bring this to the market in the appropriate way. So this is, of course, a very good example, exemplifies what the European Innovation Council is supposed to deliver. I will not go back in the details that the Keith has explained in the positioning of the EIC that is very different from other instruments that exist in the chain of research and innovation instrument. We just would like to highlight here that we are, of course, here your partner throughout the chain in order to make it work. And of course, we are with the different teams that are managing these different projects in the different instruments at your disposal in order to make this a success with the tools that we have available that give us a lot more flexibility in order to address your requests. For example, to pivot, to reorient projects, to discuss with us how we can make sure that the financial support that we are giving you is targeted. We have also a range of other tools that we put available in order to give you access to different talent pools, coaching, mentoring, financial support, and the other type of activities that we make available. And that, I think, is very essential in order to make all these possibilities a success of your initiatives. So that's what I wanted to say. Thank you very much.
We are currently, thank you, Stefan. We are currently at the stage where we are working at portfolio level, and we're all learning from this process. But this cannot be done in areas of cell and gene therapy without having very experienced people helping us. Hans-Peter Kim, Professor Kim, has been advisor to me in this field. I'd like to invite him to share his views. And I just want to say that he's what I would call a world-class scientist physician. He has been funded for 25 years, constantly from NIH. He has a tremendous track record, vast experience in the field, and he's currently, he's currently the Stephanos Family Endowed Chair for Cell and Gene Therapy at Fred Hutchinson Cancer Center. He's also directing programs of cell and gene therapy there. He's also professor of medicine at the University of Washington. He has served as an American Society of Cell and Gene Therapy and member of the American Society of Hematology, stem cell committees, and also chaired those committees. In 2022, he was served as a president of the American Society of Cell and Gene Therapy. Hans-Peter, it's a pleasure to have you with us.
Well, thanks so much, Iordanis, for this very kind introduction. It's really a great pleasure to be here and talk a little bit about my research and, of course, my vision probably and the shared vision that I think I've been discussing with Iordanis now probably for the past year now, and of course my passion for advancing cell and gene therapy in Europe as well. So I picked this topic. I've talked about this at the ESGCT meeting already, but this is going to be just a summary of an area I think that hopefully we can move into in the future. And I can see some of these portfolios already geared towards this area as well, especially our portfolio number 2, improving effectiveness and lowering the risk of gene delivery, because it's all going to be about delivery, gene delivery, as you will see. So for hematopoietic stem cell gene therapy, you can see there's obviously really thousands of genetic diseases and really millions of people are affected. So we could treat really a lot of people with gene therapy. And this is right now focusing on stem cell gene therapy, really that we can target a hematopoietic stem cell. And of course, we've got now approved therapies, and there's many stem cell gene therapy trials, but of course, all of those are with ex vivo gene therapy approaches. And even in the United States, only a very small portion of patients could actually access ex vivo gene therapy therapies in general, whether it's editing or lentiviral based therapies. So, and of course, those therapies will be very difficult to then further make them available in countries or areas in low and middle income areas, as I have shown here. But the same as the problem we have in the United States would also apply to Europe, of course, with these ex vivo gene therapies. The access would just be very limited. And this is mostly based on this here, because currently the way we do this is, of course, it's still like a bone marrow transplant, a full bone marrow transplant. So there's conditioning involved. There's, of course, manufacturing of the cells involved. And of course, this can all be improved. And this is, I think, what your portfolio 1 will accomplish here. So there's still a lot of improvements we can make here. But it's still a long process, as you can see here, to really bring this gene therapy to patients. So really what we've had that vision for quite a number of years now. Could this just be done also in vivo? And could we deliver a vector to the stem cells directly in patients? And then hopefully avoid any pre-toxic or toxic pre-treatment and really simplify the process and have an off-the-shelf therapy. Ship the vector or the lipid nanoparticle or other nanoparticle to the place and simply inject it. And what we also work on, and you'll see there's been a lot of progress in that space, to then hopefully also further enrich after we hit sort of, a certain fraction of stem cells in the patient, but be able after that to enrich and bring these editing or modification levels into a therapeutic range. So these are some of the, I think, important considerations. Of course, we want to hit the right cell. Then we want to be able, as I just mentioned, to further enrich so we don't have to come back over and over with the administration of the lipid nanoparticle or viral vector. And then again, what is going to be the best platform. So we have focused, as you can see here, on a particular population that's called CD90 population, and we've done some non-human primate studies in this space, and this just gives you an example how we can examine these studies at least in academia in non-human primates. And what we've shown here in these studies is that actually when we just sorted for this CD90 population that you can see, if I have a pointer here, I'm not sure that the pointer works. Oh, here, it does actually work. So this small population here, this is only about 5 to 10% of the overall CD34 cells. We've marked this with this green, and what was really surprising to us is this one example. When we then, again, transplanted these cells, all the reconstitution came from this very small population up here, and also all the other lineages. So there's certainly one population, CD90 positive cells, that could be targeted and again provide all the different cell populations after transplantation, and again early and late. And we've extensively studied this particular population, but as you know, there's also other stem cell populations, other investigators target, for example, CD117 or other markers. So we've just been researching and studying this particular CD90 population. Now, in terms of then, how could we enrich then for these cells? Because when you do in vivo gene therapy, you will only hit a certain fraction of cells initially, maybe 5%, but that's not going to be enough to cure sickle cell disease. So you want to hit a large enough fraction of stem cells that you can then expand these in a polyclonal manner. So I mean, you really have, certainly a polyclonal population that will contribute to the overall repopulation of cells and then contribute to the therapeutic benefit in patients. So this is why we think we will need this sort of ability to edit cells, use base editing, multiplex base editing, so we can incorporate, for example, a therapeutic edit here for fetal hemoglobin reactivation, and then a selection gene here in the same cell, so we can exactly accomplish this and then bring the levels back up into a therapeutic range. You can see you start maybe with 5%, and then depending on the therapeutic level you need, titrate it up in patients. And we've actually done this, again, with this fetal hemoglobin reactivation edit here, and then CD33 up here. With that, we've shown that we can actually nicely add these cells engraft in mice, and we can then actually protect this population here with an antibody-based treatment and also select in vivo. And there's already some data out there that we've that are in bioRxiv right now, and then also the CD33 knockout approach is actually already in the clinic for an allogeneic transplant approach by the company Vor, to then be able to give, in fact, after transplantation, either CAR T cells or an ADC targeting CD33 cells to get rid of leukemia and then protect the modified cells. And the same concept we can use again to then select for these modified cells in patients with a double edit, for example, for fetal hemoglobin reactivation and then that CD33 selection gene. And here are just some of these others. We're not alone again, and this is what's so exciting. So we're not alone. This is not like one lab doing this. There's Beam working on this. There's other people now, Pietro Genovese from Boston, Harvard, and Dana-Farber just published this. And of course, Lucas Jaker here has just published this, and Chimeo. So there's really a number of people in labs really around the world working on this right now, and some of them also coming from Europe to really pioneer this approach. And then, of course, the delivery. And I mean, Drew, of course, now got the Nobel Prize, but he's really pioneered the lipid nanoparticle delivery. And that's, again, been a recent paper for in vivo gene therapy for hemoglobinopathies. So again, this is coming. We've worked on other nanoparticles, for example, polymer-based nanoparticles, or we've also explored the gold nanoparticles. But what you see here is actually viral-based delivery. This was pioneered in Europe. All these investigators are actually from Europe, and this started out here to really start in vivo gene therapy for stem cells and for T cells. So all these things have started actually over here to a large part. So we have also worked with the viral vectors that we can, similar viral vectors that you've seen before that were used by Els Verheuyen, that we can now modify and actually put a CD90 targeting scFv on outside of the lentiviral particle to specifically target these CD90 positive cells in vivo, in mice, what we've shown that. And again, this is, of course, again, the goal for all these different approaches targeting these different antigens. Of course, there's another approach with an adenoviral vector system. This has been a collaboration with my colleague in Seattle, Andre Lieber, using a helper-dependent adenoviral vector system that has a modified fiber, that Ad35++ fiber, which will actually target very efficiently CD46, which is highly expressed on these CD90 population I've been talking to you about. So these are just different kinds of vector systems that we can use now for this in vivo delivery. And with this vector system, we've also shown that we can deliver a fairly large prime editor to fix sickle cell disease in a mouse model. So I think there's lots of very promising data out there right now. And I think I just want to move to this slide here. And this was last year. Francis Collins gave the keynote, one of the keynotes at ESGCT. And the question was, of course, why in vivo, why gene editing? Collins asked rhetorically as he returned to his central theme, there are thousands of rare diseases. I want to find something that's scalable. And that's exactly what this is all about. And that's it. Thank you very much.
So our focus is scaling up, and we are really fortunate to have Hans-Peter advising us. At the portfolio level, the projects, they are very much concerned, of course, about the future and establishing partnerships, and they really want to know what is in the interest of the big industry. Our second speaker comes from the big industry, and he has vast experience. He has worked in many different pharma, and he has a big track record in RNA therapeutics. It's my pleasure, great pleasure, to invite Dr. Michael Wenger with us to share his views in the field, in the revolutionary field that he works for BioNTech, and we have a long lasting relation with BioNTech. We previously organized meetings on licensing with my colleague Anne-Marie Sassen, who sits over there from EIC, and we had Chris Vogel, but now we have their Vice President of Clinical Development, and We're very happy, pleased to have you here.
Thank you very much. I'm not sure I would qualify for a company that is big pharma or large pharma. BioNTech pre-COVID was less than 1,000 people. We've grown a little bit since, but we still consider us very much a biotech company. That said, due to some of the success of COVID, we're now able to conduct a lot of things that our founders were actually thinking about, together actually with the other Nobel Prize winner, Katalin Karikó, who worked at BioNTech since 2009 for about 10 years. So basically the company was founded as an oncology company. You might know BioNTech as a COVID company, but it was really founded as an oncology company based on the mRNA principle. And mRNA was really what the founders wanted to bring to patients, and it took them, give or take, 10 years to develop the technology, to fine-tune the RNA, to put it into the lipid nanoparticles, and then COVID came along and the technology was ready. But it's only one part of the story, right? The other is cell and gene. I'll talk a little bit more about this. But a large focus of BioNTech is also protein-based treatments, normal antibodies, but also mRNA-based antibodies. So we inject the mRNA and the body builds the actual antibody. And ADCs and also bispecifics. And then we have a small part of the company that deals with small molecules and many more things. What I'm telling you now about are only the projects that are in the clinic. So we're moving gears a little bit also from the portfolio aspect. But if you bear this in mind, we also think portfolio. We also think portfolios have an economy of scale. So we can't do everything, even if you're richly funded, which most of you unfortunately are not. But if you're richly funded, you still need people, you still need time, you need brain power, and so we need to look portfolios. So this is our pipeline part one that we have in oncology. There's a similar slide for infectious diseases that I'll spare you. We do have off-the-shelf vaccines, quite a few actually. This list is not complete. So mRNA vaccines that are built around a particular tumor entity where we inject the mRNA based on antigens that we believe, a number of antigens that we believe are not modified or changed during tumorigenesis and are collectively expressed to 90% plus. Then we have the individualized vaccines that also the colleagues from Moderna are developing. You probably heard these stories. Then there are intratumoral immunotherapy that has become a bit out of fashion, including with us. There's ribomaps, I alluded to this. We inject mRNA and build the antibodies. There's cytokines like interleukin-2 or interleukin-7, or a variant of IL-2 that we also build by injecting RNA. And then the field of cell therapies, we do CAR T cells, and you'll hear about this in a moment a bit more. And we have neoantigen-based T cells and actually a large number of projects in preclinical development. Again, everything you see here is in clinical development already. Then checkpoint immunomodulators, a project that we're co-developing with a company called GenMap, and some simple antibodies, and a few more things that I don't need to talk about here. So basically, I thought it might be fitting if I show you some of the data that was presented just a few days ago at ESMO for a combination of a vaccine, which is kind of a gene therapy, with CAR T cells, which obviously is cell therapy. This was presented by John Hanen from the Dutch Cancer Institute, again, just a few days ago, I'm sorry. And I'll just give you a brief tour de force over his talk here. So basically for CAR T cells, and you heard several aspects of this during this meeting, there's a number of problems in solid tumors. You heard CAR T cells in hematology. They are out now for 6-7 years or so. They work beautifully, but it took a while to understand them. And having worked at Novartis, we also had a big long learning process of scale up, of going from manual to automated manufacturing, etc. But why don't they work so well in solid tumors has been discussed many, many times. It's basically 2 or 3 things. Number 1, there aren't any really good targets. Number 2, the microenvironment basically is a problem for getting the cells to the tumor. There's issues with persistence of the CARs. So claudins are a group of targets that have been discovered only a couple of years ago. Also, Özlem Türeci and Ugur Şahin were involved in this. And the good thing about claudins are that they are silenced during organogenesis, right? So they're basically not present in healthy adults. But they're expressed in a variety of tumors, and then you see here we can make both the CAR-T and we can make also the CAR-VAC. Why do we need the CAR-VAC? Well, the CAR-VAC is basically the idea of stimulating existing CAR-Ts to multiply further, to basically stay longer and also expand during the process. It's something that we thought we can do as a company. We didn't know if it works, and even if it worked, we didn't know if it was actually necessary. So because of this, we conducted a phase 1, typical phase 1 study. You see here, I'll spare you the details, and had around 44 patients. By now it's a few more treated with this. Most of these patients were in testicular cancers. Second largest group was in ovarian, and there was a splattering of other cancers. Heavily pretreated, as you can imagine, in this setting. And from a safety perspective, turned out that the CAR is more or less similar to several other CARs. We didn't see many concerning things, but we did see the usual CRS, usually grade 1, 2, but there were a few higher grade cases. For the CAR-VAC, similar to what you would expect from an mRNA vaccine, you get flu-like symptoms, but nothing really concerning. Now, does it actually work? Well, the efficacy from those that from the data that we have in all dose levels looks pretty encouraging. If we go to the higher dose level, we do see quite nice responses. And they're also, as you see on the spider plot on the right-hand side, they tend to be durable. Now, the interesting piece is this CAR-VAC, does it actually work? And that you see here. On the left-hand side, there was a cohort of patients that did not receive the CAR-VAC, and what you see are the kinetics over time of the persistence of the CAR-T cells. And on the right-hand side, when we boost the CAR-T cells with injection of the vaccine every 3 weeks, they do tend to live longer. So I'll save you the summary for time reasons, but I wanted to again come back to the point of portfolios and why you're here. This is a clinical example of a company that was obviously, or that is obviously successful and that had enjoyed a run. But it's a lot of work to get here. But it's also only possible because there's focus on specific areas. And it comes from several sides. So this house was not built on one day, obviously, but it was built by very skilled but also very focused people, similar to the portfolio concept. And this is also how we decide if we want to partner with companies like yours, right? Like we're not looking for that individual thing that might have a massive improvement. It would be nice if we found the needle in the haystack, but we probably don't. So we're looking more at again, economy of scale. Is there something that might work? We might want to invest in a cluster of things rather than in a single thing. Thank you for your attention.
Can't be more reassuring to listen from you that you also have interest in portfolio and the very targeted approaches, Mikael. But the portfolio, in order to establish a portfolio, you need to select the projects. And in this process, I was very fortunate when we did this work at the EIC 3 years ago. In the panel that selected the projects, the 9 projects that you are here today, was Professor Seppo Hirtuala, who is a world leader in gene transfer. As far as I remember, in the 90s, you were the first that transferred gene in the arteries. And he is with the University of Eastern Finland. Has a lot of experience and success also in bladder cancer oncology, and he was, I believe, past president of the European Society of Cell and Gene Therapy for 2 years, and chief editor of the Molecular Therapy. And his company, the company that he participates, they got an approval for bladder cancer from FDA 2022, which is obviously a big success. And with these comments, I'd like to invite Seppo to the podium. It's a pleasure to have you here.
Thank you. I'll be brief. I don't show you any real data, but I give you perspective as a reviewer and European scientist who wants to find funding. So just to give you background, so I do have quite a lot of experience from ERC grant reviewing all these different grants, Marie Curie programs, EU EATRIS Infra funding, Videra funding, and then of course all the European Union Horizon type programs. And the panels I've served as either member or chair are mostly Life Science 7, co-grants, synergy grants, and now also the IIC panel. How do we know, we who do the research and compete for funding? I think this instrument has a very good reputation among us who apply these all the time. Excellent grant system. And I think that what we've heard here, how do we support EU science, innovation, and technological autonomy? This is a new tool for us. You all know about horizon restrictions, and then all these even synergy grants tend to end up in situations that groups work together and continue what they've done in the past. And I think that in this sense, this was very refreshing and new also to me to be in the AIC panel. And it clearly complements Horizon and ERC grants. High quality proposals, most of them had to throw away because just didn't have enough budget. So I would like to see 1-0 at the end of the amounts that you have in the brochure. How much you give 3 million, I would like you to give 30 million for programs. Very low acceptance rate, as you might know. And what happened then in this review system? You have a room full of people with quite big egos, and then you think about what to do with these grants. And so it happened that actually proposal came from program manager. Why don't we put the best projects in a certain key area in the same basket? I found it wonderful that even though you had to sort of throw away some very, very good high-scoring grants, but they were not fitting into the, I would call it pipeline, or we call it portfolio here. And I think that indeed in the European Union system, this was excellent, visionary, and accepted after a little bit discussion by the panel. I think it's a major development in European grant system that not only you have the high science score, but then you have something else, and then you force the people to work together. I mean, the different grants, as you've seen here in previous talks. It allows us to build up critical mass of researchers in certain key areas. Of course, the key Achilles tendon is what areas do you select, but that's then, of course, a process, both in European Union in these committees, but then also our scientists, we can contribute and give you ideas. And this should really speed up development and applications in selected topic areas, and indeed improves collaboration beyond single grant consortia, because most of you know that it's so easy. You got your 3 million or 6 million of EU funding, and then the groups just for 5 years, and then they keep doing the work, maybe they share a mouse model and so on, but that's it. And I think that has been, even after getting 20 different EU grants worth quite a lot of money, that's always disappointing that what's written in the original application may not always come through. So I think this is a real advancement. And I just the last slide, so as Iordanis just mentioned, so finally we got approval from FDA for our bladder cancer product. It's adenovirus interferon alpha called adstiladrin. It's put with a catheter in the bladder. And indeed, we had to go to US for the final studies because of the funding and getting things working more properly. But what if, because it took 15 years to get this approval, what about if we had had EIC at that time? That might have been gone much faster and might have been able to do that in Europe. So I think that in this new grant program, there is an opportunity, as you heard, also beyond science, but going to translation and manufacturing and clinical trials. Even company can get funding. So I think this is fundamentally different, all in the same baskets. So as you can see here, so all these aspects are needed, not to forget regulatory aspects, which are formidable both in US and in Europe. So I think this has been a great development, and I really want to see what are the next key topic areas that will be funded through this system. So thank you.
Great words by Sebo. Thank you very much, Professor Hirtuala. Thank you for being with us when we selected the projects and sharing the vision. I think now it's the time to see how we work at the portfolio level, what is the real interaction between the projects, and I'd like to invite to the floor John Hodgson, former editor-at-large with Nature Biotechnology, currently contributor and advisor of the same journal, to chair the discussion between the projects that we have at our portfolio. And John will take it from here and then introduce the projects. Please, all panelists are, please join us.
Okay, good. So we have to get into this very fast because one of our panelists has to leave, which is a shame. Very busy man, Alberto Riccio. This is the nitty-gritty of what it's really like to work in a portfolio. At least I hope that's what's going to come out. So I'm going to just introduce Alberto first and then ask him a question, and then we'll go on to the rest of the panel, if that's okay. So Alberto is the professor of the molecular therapy program at the Telethon Institute of Genetics and Medicine in Italy, and vice president of the European Society of Gene Therapy and Gene and Cell Therapy. So before you go, Alberto, Danos mentioned the sort of three areas that are covered in the overall EIC program. So there's the technology, there's the regulation, and then there's a sort of commercialization or exploitation. Can you talk briefly about your experiences in all those three areas? And I know it's going to focus on the adenovirus models.
So the experience that we had so far in the sub-portfolio number 2 are mostly around tech, almost exclusively about the technical aspects. And there are a number of collaborations for different tasks between our consortium, Evolution, and the other 2 consortia that are Novistem and EDGT that are in place. But this is all science. And what I noticed, and I think this is important, is that within the portfolio program, there is also 2 other type of activities that are exploitation and regulatory aspects that we didn't explore yet, but I think are very important. And typically these aspects are actually things that scientists look at later stages, once proof of concept has already been established and something is moving towards translation. But I think these sub-portfolio activities give us the opportunity to look at these things early on, which I think it's important because, I mean, even basic discovery can take advantage of this type of interaction that can shape how then the further step of research will go.
So you haven't done that step yet, but you think
not
it's
those
going to
two
be...
aspects, not yet.
Have you got in mind...
We have the opportunity to do it with the
right.
EIC portfolio activities, and I think this is important, as important as the...
Have you got any specific questions in your mind that you would like to address to the kind of advisors that are available on the regulatory side or on the commercialization, exploitation
Well, I think
side?
the regulatory path of some of these activities should be known, and so if we know it from the beginning, this could help us design our experiment, even our early experiment, in a way that we're going to gather information towards even regulatory goals, and the same goes for exploitation, like the IP generation and future further exploitation through spin-off that can come later. So this can all be discussed at an early stage, so that we are at least aware of what are the...
And it's going to reorientate your research program in a research and development program in a way?
Yeah. Well, I think particularly for this type of research, this would be the final goal. So, yes.
Yes. Do you really have to go right now?
Yeah,
I think you do. Okay, fine.
I apologize, but
thank you so much for that input. I think we have some experience, more direct experience of actually using those mechanisms from the rest of the panel. So thank you, Alberto, for staying for this precious few minutes. So I'm going to introduce the rest of the panel now. Thank you. No problem. So briefly, and if I put my glasses on so I identify the right people, starting from this end, we have Mariana Werner Sunderland, who's a senior scientist at Achilles Therapeutics, and they're developing, well, early stage of developing neoantigen targeted T-cell therapies. Next to her is Luca Gattinoni, and he's professor at the Leibniz Institute of Immunology, formerly the Regensburg Center for Interventional Immunology in Germany. And then next to him is Bernard Gentner, who is the professor and a physician scientist at the Department of Oncology at the Institute of Cancer Research in Lausanne, and was a founder, I believe, of Genenta, which is now a NASDAQ-listed, US-listed, but basically European company listed in the US. And furthest from me is Luc Henry, who's CEO and co-founder at Limula, which is based in Switzerland, and they're developing an automated modular manufacturing system. So you'll see how some of the component parts that our panel represents fit together. So just to pick up on the point that Alberto was raising there, I know, Mariana, you have some experience with, at least on the regulatory side, with the project. So say something a little bit about the project and what you were looking for, and then how that came into play, if you would.
So, yeah, hi. So I'm representing the Smarter Consortium, and so we had early on a webinar that was organized by our program managers, Iordanis and Ioannis, with EMA experts, and we thought that it was very useful because, as my colleague was saying, there are certain things that you usually don't think about in regulatory terms until you're way down the line, and by then it's usually painful to have to go back and actually reorganize things or change things to kind of jump through certain hoops. So I went to this webinar, and the information that I got from that, from the experts, was also because it was very tailored for the CGT field, and what to expect in the framework going forward. And that was really useful for me to bring back to the Smarter Consortium, to be able to kind of think if what we're doing fits what we would need to do in regulatory terms later on.
So how was that meeting set up? How was that webinar set up?
So it was an idea from our program managers to, instead of having how these EMA experts usually have these meetings, but it's a more broad subject, our program managers decided that it was a good idea as a portfolio activity to have it specifically for cell and gene therapies. So then all of the information that they gave us was very useful for our field specifically. So that was the benefit, one of the benefits of being in this portfolio and having this connection.
So they essentially connected you with the precise person, not a general inquiry at the EMA.
Yeah, exactly.
Okay, thank you. So I think, does anybody else have any sort of inputs that relate to the regulatory side or to the exploitation side before we move on to something else that were elements of, part of the portfolio approach? I think Luke has something to say.
I mean, at the moment it's
Sorry, I've gone off script, so everyone's confused.
So I think at the moment for our project it's still a bit early to talk about regulatory issues. So they will be coming up for sure in the next couple of years. So I'm looking forward to additional activities in this area, certainly.
So you say that from the voice, as a voice of experience, because you know these things happen with your experience at Genenta. Is there a comparison, Bernard, between the sort of activities within the portfolio or within your project and the pathway that you'll have to follow because of what's happened in the development of Genenta's product?
So I think we're going here even a step further, because let's say the companies usually they take a standard approach, which has been already tested in the clinics. But here, I think the ambition is to go beyond the standard and to innovate the way how we perform gene therapy, and this raises regulatory questions which are new. So I think this goes beyond what we have done in the past and requires some specific questions to be addressed.
Yeah. Just come back to you, Marianne. How early in the project was this? Just to pin this down. Sorry, I should have asked that when you were...
No, it's fine. So we are still in the first year. We just finished our first year of the project, and this webinar took place, I think, like a month ago or something like that.
So it's first year of how long a project?
Of 3 years.
3 years, so really early on. Then.
Yeah, but I think that if I can say another point is that it's really useful to have these kind of more tailored webinars, because then it also gives us kind of, the academics and the industry members a say in how this framework will develop in this field. Because I think having, this opportunity to talk to the EMA experts and kind of say like, well, this is what we are kind of doing, and how is the regulatory framework gonna change with these new innovations?
Yeah, you can't say anything specific about how those discussions have influenced your program.
Sorry.
Okay, that was a VACO, but keep the microphone for a minute, because I'm going to stick with you again, because the Smarter project also had interactions with other parts of like on the manufacturing side, I believe, and that was something that apparently you found useful. Can you just sort of walk us through that?
Yes, so one of the experiences that we had in the Smarter project as part of the portfolio was, which I think was one of the most interesting and valuable experiences, was the fact that we were able to talk to all of these lovely people here and see all of the different projects that are in the portfolio and try to find any kind of crossroads where we could interact and we could align and collaborate. So we had a lot of one-to-one conversations with all of them. And because of being in the portfolio, we were able to actually, we talked to Limula, to Luke. So he's the technical lead of Path for CGT. And we realized that there was a very good chance for us to be able to collaborate because of a model that we had developed in the Smarter Consortium and a technology that that Luke's company does, the bioreactor that they do, that they were using for the Path for CGT. It just made sense for us to trial that technology with our model and see if it worked as well. And this is something that we wouldn't have been able to do if we were in the portfolio, because, to be honest, we didn't have Limula in our radar. So it wasn't something, it was something that came up directly from being in these meetings. And we are going to start this collaboration early next year. So we're really excited to try out that.
So just to pin that down again, if I can, it occurs to me that there's nothing stopping you ringing Luke or anybody else up, or any of the panel, outside of EIC, not funded by anybody. What is it about, I'm just saying, can you pin down what it is that within the program stimulates that activity?
Yeah, so I think that it's the fact that we can, just by being in this program and having this kind of different projects that are aligned in a way towards the same goal, you can double the investment in a way by collaborating, by something that we are already doing. It's kind of like parallel thinking in a way, and something that we were already doing for a project, just extended a tiny bit more and use some of the things that the other project is doing. And then, I guess that I would put it as like the sum of our parts is just bigger than our individual components, if that makes sense. And that would be something that if we had gone a different way or a different grant or whatever, we wouldn't have had the opportunity to do.
But is it kind of like you're locked in a room with these people and you're forced to talk to them? Or
Well, I mean, maybe at the beginning it was like that. But no, I think that it was because all of our projects have something that they can align with, it's just easier. If it was something that, it came from completely different places, then it wouldn't be useful. But a lot of these projects, you can see that there is alignment there.
Right. Luke, I'm going to come to you next, actually, because I think it seems to me that, particularly within the context of this conference, automated methods for cell and gene therapy production or manufacturing are going to be, key to everybody. So was it your role, or was it Limula's role, essentially, to, provide the solution to the problems that people are having, or was your involvement somehow going beyond that and benefiting from interactions with the other people in the group?
Thank you. It's a good question. I think, first of all, I need to acknowledge that there's a lot of innovation and progress made in automation for cell and gene therapy beyond what we do. We're trying to bring a next generation smart bioreactor to the market, and we're relatively early stage, so we still have room for adapting, improving the technology in the next 2 years. And so the main value that we see in this consortia is not just developing the technology in the portfolio, in the consortium that we've built, but also being exposed to other end users, other types of potential users. And so the value we saw was being confronted or having to explain what we're trying to do to people outside of the proof of concept scope that we had initially in mind. So we've been working on CAR T cells, on T cells, modification and expansion exclusively, because that's where our expertise is. But by showing this technology to people working in very different fields, suddenly we realize, okay, there's also an interest in these other cell types. And so in the TILS case, and also with Bernard here on the hematopoietic stem cell space, suddenly we would get feedback that we wouldn't have been looking for necessarily.
So it's not just a question of like beta testing your system on different cell types that are brought to you by these clients that are paid for by the EIC, or is it more than that?
So the beta testing was planned as part of our project, but exclusively on CAR T cells and CAR NK cells. So I think the value we have in these exchanges beyond the consortium is in understanding the requirements from people in a different space. And so understanding what is the versatility of our tool. Can we actually, without making too many technical changes, address problems in other niches of cell and gene therapy? And so this inputs into the design of our technology will hopefully allow us to expand the scope of its use.
So you now know that you can adapt your system so that it works with a wider range of cells that you hadn't tested on before.
So the hope is that we don't need to adapt it,
okay.
but that we, build it the same for all the applications, because again, we're still in a position that we're taking in user requirements to build what would be the final version. And so the more diverse inputs, feedbacks, and requirements we collect, the more we can see, okay, what is common between the different areas, and how can we take them into consideration.
So up to the point where you were involved in this project, had you just seen a range of standard cells, or how did this take the company forward in some way?
I mean, clearly, as a small, early-stage company, you cannot build the know-how in-house to address every possible applications. And so beta testers within the consortium already bring a lot of knowledge, but again, they may not be experts in all of these cell types. And so having the opportunity to conceptually first and then experimentally test the technology in the hands of multiple players is absolutely key to validation and exploring that scope of use.
Thank you. Luca, I'm going to come to you next, partly because I kept you quiet. But when we were talking earlier, you'd mentioned what I thought was a great example of one of the challenges that is facing a range of next-generation gene-edited therapies. So give something about your background and the fact that you're approaching this through the gene editing route, and tell us what you learned as part of the crossover activity here.
Yeah, I'm representing T-Fitness, and during the brainstorming session, I think it came immediately clear potential nice synergy with the EXPAND consortium led by Bernard here. Because we both focus on a platform that is rely on CRISPR-Cas gene editing system. And as a consortium, we mostly focus our platform on electroporation-based system and way to engineering. And this can be problematic, especially when you work with T cells and hematopoietic stem cells, because by doing this, you have to pay a lot in terms of cell yield due to the high mortality of the electroporation phase. And we know that then by being forced to expand the cells in vitro to reach a therapeutic relevant number, then you can lose stemness. That is a key both for hematopoietic stem cells and T cells. So if we find a way to preserve the yield after performing a CRISPR-Cas editing, I think is important. And talking with Bernard, we discussed about the potential of using the lipid nanoparticle technology that they were also developing together with Luigi Naldini. And we thought to in parallel test also this platform to gene edit ourselves.
So the ability, so you're developing a system which may be widely applicable. Again, it's this sense of a common tool that could be used in many places.
Yeah, this is essentially the platform can be applied to any type of cells. I think we focus mostly on T cells because is what our consortium is doing is generating a better T cell product. The focus of EXPAND is hematopoietic stem cells, so they will focus on HSC at the same time.
Right.
But this potentially can be used for NK cells and any other type of cells that can be that you want to engineer.
I'm going to open the floor to questions in a minute. Just if you have any about future programs and so forth, we have various people in the room who can answer them. But first of all, I would just want to ask the panel who've had experience of essentially the first version, the start of a new type of R&D funding. It was a relatively new format. I guess elements of it are elsewhere. But were there sort of difficulties in bedding the system down? working out how the projects work, how the portfolios work, were there teething problems? And if there were, how were these overcome? What are the learnings, essentially, from your first run at this in the cell and gene therapy area of the kind of portfolio approach and the projects involved?
So what I wish would have been clearer since the beginning was that this idea of portfolio activity should be presented already at the time of the call. So it was kind of a surprise to us that we were informed during the grant preparation that we have to divert part of the funds that we were applying for our project to support this portfolio activity.
Okay, and I thought there was so much discussion here of portfolio, I thought it must have been overt at the beginning, but...
No, that one we will learn in itinerary, I will say in, in Latin. But and so I think that this could have created some potential missing opportunity because we could not move too much of our planned funds from our project to this activity, so we limited the amount of funds that were moved to the portfolio activity to 1.5% of the budget. So it's not a lot. It's enough to explore parallel activity, but not to go far away from what you have originally planned. So you can see some benefit, but potentially if we had more funds or
just
we knew more, we could have create or brainstorm even bigger ideas.
So 1.5% is what you've allocated?
Yeah, this is from my budget, yeah.
Do you have a sense of how much would have been useful? 10 times that? Are you using the zeros that are floating around?
Yeah, now that depends on the discussion because now we were already aware of the constraint of the budget. Also, when we discuss between each other during the brainstorming session, we already know that we could not aim at something huge, but you can aim at something that could implement and boost your portfolio, your project, and so we were also not discussing bigger ideas.
All right.
But
hopefully
potentially, I think we'll be interested.
We'll get some input from Danos or somebody on whether that's going to change. Any other teething problems from other people on the panel, or solutions or learnings to use the...
So I guess by design this was a black box, no? Because we couldn't know who would be the other winners of this grant. So of course you cannot really plan for portfolio activity if you don't know what are the projects which will be funded. So we couldn't know the topic that would be underlie this portfolio. So this was a bit of a surprise, of course, no? Then we spontaneously found interesting collaborations, interesting projects. And so with the budget that is available to do activities, this is enough for a proof of concept, right? So to understand feasibility. If you do more ambitious collaborations and developments, this would certainly require extra funding, which is at the moment not foreseen, but which could hopefully come out in the future. No? If for the winning collaborations, where really we see that a synergy between two different groups is developing.
I got one cheeky question, which is, was the fact that the Pathfinder is sort of a route into the transitional and accelerator phases, greater funding or extended projects, was that an incentive for you to get involved in the Pathfinder itself, or is that just a useful bonus?
So most definitely it's an incentive.
And for us specifically, because to clarify, so the Pathfinder CGT project is focusing on developing sensors to monitor cell culture in the bioreactor. So in essence, the funding is not supporting the core activities of the company. It's developing a must-have now, I think, functionality to that technology. And so without that very early stage kind of moonshot funding, we as a company would not probably spend a lot of R&D on what has proven to be a must-have functionality. Yeah. So I think it triggered a new activity and a very valuable feature to our product as a side track that we'll incorporate eventually.
I think moonshot, maybe pushing the budgets a bit for cell and gene therapy, but we'll come to that in a minute. Are there any questions from the floor? Because we have an experienced panel of people who've gone through the process. Maybe some of you would like to get involved in the future. Yes. Hobble down or shout it out and I'll repeat it.
Thank you. It's half a remark and half a question. So, for the
make it mainly question, please.
Yes. Rebecca, head of Transition Business Acceleration Services. For the complementary activities, whether about the portfolio or even for exploring potential innovation, we have now the booster grants, okay, which are not huge, 50,000, but you can get on top of what you have. So my question is if you are aware about them or you thought about applying for them, because we already have the first application, so this is up and running. Thank you. So how about booster grant? Did you thought about it?
I'm not aware of this myself,
Yeah.
so...
I was not aware. What actually we explored was the hop-on possibility. So with extending the consortium to partner from a widening country, so called Define. And so we interact with Roman Gerala from Slovenia to enlarge the consortium, and let's see if we get funded or not. So we apply in September, and hopefully this will help it.
Sorry, I am aware of the Smarter Consortium is aware of the booster grant, and it is something that we are thinking of maybe applying later, but obviously we need first to have some kind of proof. Like it's always the timings, right? So we are planning on, like as it was said, this is just like a small proof of concept, see if it can be done, and then apply for the next thing, obviously, if it's successful, which we are very hopeful that it will be. Okay, good to know, thank you.
You can apply any time, says our questioner. Next question.
Good afternoon. I'm Alex from the European Platform on Nanomedicine. First of all, congrats and thank you. We love the idea. It's new, so I guess the black box sometimes might be a bit surprising. So my question may be a bit tricky, but how would you include that spirit in future calls? What would you, because we always say we don't want to reinvent the wheel or to have mega overlap with other projects. So between overlaps and synergy or portfolio spirit in the future calls, how would you like to include that spirit, if doable? I know.
Tricky question.
So the question is whether one thinks about restraining further the topic of the call, maybe, no? With cell and gene therapy can be very broad. It can go from manufacturing to, to many different other things. So restricting it further could potentially make it more easy to find, a portfolio. But it comes also with downsides, of course, no? So if you're more restricted, you may not get such a huge variety of proposals. So what I would think would be positive to have, for maybe not for all portfolio interactions, but for the ones which are the most promising and extra funding already integrated into the call, which can be triggered after a proof of concept. That would be helpful, I think, to promote the winners, I think, of this.
So the balance seems to be about keeping it narrow so that you can have a self-defined group of things that are likely to interact, or casting the net wide and then disappointing a lot of people because they don't fit into a group. Is that the challenge, Danos?
Yes,
Tell us.
I think there are two different things here. One is how you select the portfolio. You try to maximize the common element between the projects. And that's the key to, it appears to be the key to success. Because then you have a good reason to work with other projects. You don't need to be convinced, is what Luke just described, and Bernard, they wanted to test. They were really close subject-wise. Then if the concern is whether you should go really very targeted and that you're going to miss something, I believe the future is going to be really very targeted approaches. And that is not something we can influence right now. And the idea of developing strategic autonomy in Europe, it can, in my understanding, can happen only in highly selected targeted areas. Because the portfolio could be the nucleus. In this case, the portfolio is at the very early stage. Could be a portfolio at the transition level. or accelerator level. These are things that we are testing and we're going to develop at the EIC. Around those, you bring other projects in a little bit broader area of cell and gene therapy that always you apply the same logic. You need to have lots of commonalities. I'm presenting the investment of 200 million by Ohio Discovery Institute to Oxford University, which is very successful, for the sole purpose to develop programs in rare diseases using cell and gene therapy at the accelerator level. And they realize now they need to go even more targeted. Have I answered your question?
Thank you. Thanks, Dennis. So we're probably coming towards the end of our time, but before I let the panel go, one of the things that struck me about this program was the kind of active management, the program manager role and so forth. And I think my impression from what you've been talking about is that has achieved things. It's locked you in a room, it's forced you to talk to each other, talk to regulators, perhaps talk to some exploitation, people involved in the exploitation side as well in the future. So as a kind of vote of confidence or not in the program managers and the idea of active management of research and development programs, I'm going to give you 3 words each to summarize your feelings. I mean, they could be anywhere on the spectrum. So I'm going to start with you, Luke, if you don't mind.
I'll need more than 3 words.
More than 3 words, okay.
You know, I'm in a very awkward position because being based in Switzerland, you know, the policy aspect of working with European funding is something that we'll discuss over a drink later. But in general, I found that having a peer experts to talk to and whose interests are fully aligned with those of the different companies and research institutes is really helpful to explain maybe some changes in the project that you can justify, you know, based on the actual data, the science. And we've seen a lot of flexibility from the Swiss funding agencies where you have these interactions. They're very productive, and it's really nice to see that the EIC is putting in place similar channels for conversations.
So your 3 words are Swiss idea first. Bernard.
Okay, my 3 words are stimulating, flexible, but not coercive.
Okay. You can have more than 3 if you want to explain, but it's up to you.
Yeah, no, so it's important to keep this open and not, you know, you cannot force people too much. They find portfolio activities when they see a value, you know, and they like collaborating with each other. This is very important. So it must come from everybody. And people must do that because they enjoy doing it, not because they're forced to do it.
I think the idea of running portfolio activity is a wonderful idea. And I think that there is also a great room for improvement on the platform. And I look forward also for future opportunities and with different calls.
Improvement through meddling, then.
Great first trial.
Okay, experimental reluctance.
I mean, we are the guinea pigs in here, but I think that it has been a great first trial. I think I can speak for everyone in this panel about that. Yeah.
Okay, last chance for a question from the floor before I close the session and thank our panelists. Okay, please chat amongst yourselves, come up and talk to the panelists. I'm sure they've got something to share.